you very much. First of all, thanks to KARIM for the kind invitation to present some of my work here today or this morning. It's always a pleasure to visit Paris in the spring. My name is Professor Frank Martin. I'm based at Lancaster University. I hold a chair in biological chemistry, and I'm also the director of the Center for Biophotonics based at Lancaster University. This morning I'm going to tell you a little bit about my research interests, some of the facilities that I have in my laboratory, and an example of one particular industrial collaboration that I've developed over the last number of years. I've got a number of research interests. I've got actually quite a broad portfolio of things that I've looked at over the last number of years. Includes I have research interest in the etiology or causative factors, environmental factors in cancer and hormone-responsive tissues, primarily those of the prostate and breast, where you find that there are regions of high incidence versus regions of low incidence, and amongst migrant populations, cancer incidence changes, and we can possibly associate that with lifestyle changes or environmental influences. I've done a lot of work over the years on the development and the application of short-term genotoxicity assays, primarily the alkaline Comet assay and the cytokinesis block micronucleus assay, a measure of chromosomal damage in mammalian cells. I'm particularly interested, especially in the last 10 years or so, in low-dose effects. Consistently, over quite a number of people I've had in the last 10 years, in excess of 50 independent researchers work in my laboratory, we have seen what we attribute to as low-dose effects induced by particular contaminants at concentrations that are not dissimilar to those that you observe in the environment. And also some of these are associated with endocrine active properties. Particular gene I've done some work on and I'm interested in, especially in prostate gland, is CYP1B1, a phase 1 metabolizing enzyme. I've developed a recent interest in epigenetic effects. We've been doing that particularly with perfluorinated compounds. And finally, a major focus in my research is the interdisciplinary an emerging field of biospectroscopy, the taking of physical sciences platforms and using these to address and shed new insights into biological questions, and to harness these particular platforms with novel computational algorithms. As I said, I've got a number of facilities. I have a Center for Biophotonics. In the last couple of years, we've installed approximately £500,000 worth of microscopy and novel microscopy and imaging technology in that particular center. We do a lot of work on mammalian cell tissue culture, PCR, which is normal. We have a lot of optical microscopy, fluorescence confocal, and I also collaborate a lot with people who use and apply electron microscopy. As I said, already mentioned, we do quite a number of experiments using short-term genotoxicity assays, especially during the 1990s. I played a role in developing some of these assays, which are now routinely employed for regulatory toxicology. I also have some novel imaging kit, which includes Raman spectroscopy, ATR, FTIR spectroscopy, AFM or atomic force, and we also have access to a number of other routine methodologies or conventional methods that one might use in a particular lab. And I've got two main laboratories, which you can see from some of these photographs, a reasonable amount of bench space and laboratory space, people working in fume hood, preparing for a PCR reaction, tissue culture here, again, someone working at the bench, preparing tissue sections. I have a lot of collaborations, clinical collaborations, which I've developed and fostered over the years, and a particular bench space in my lab. As I said, I think in terms of techniques that one might use for toxicology, the two that people might be most familiar with is the cytokinesis block micronucleus assay. This is where you can take a growing cell population, you can block them post-mitosis to inhibit cytokinesis. So you can identify binucleate cells. And if a chromosomal damaging event has occurred associated with a particular exposure, you can visualize that in the form of a little micronucleus, which is what it is, a small nucleus that is present in the cytoplasm, and you can score these. I've also done an incredible amount of work on what we call the alkaline single-cell gel electrophoresis assay, or the COMET assay, is to measure DNA breaks. And essentially what this is a micro-electrophoretic technique to facilitate the direct visualization of DNA damage in individual cell genomes. And that is what it is. You take the DNA or the nucleus from a cell or population of cells, post an exposure, and under a certain number of conditions, you can electrophorese the DNA. And if there's DNA damage, the DNA migrates in a size-related fashion, which you can then use a fluorescent dye to visualize how far the DNA has migrated. And this gives rise to what we call the COMET assay, because it looks like a comet under the microscope. We also do, as I said, a lot of clinical studies. So I've had a lot of links and quite a number of epidemiology studies where we've done very robust studies, and we can prepare samples for microscopy analysis in order to, in this particular fashion, to derive a fingerprint of the tissue. And essentially, my ambition for this over the next 10 or so years is to be able to have a microscopy system based on light that can allow you to identify the tissue architecture, and inherent in that tissue architecture is the underlying biochemistry. And that has huge applications in several settings. For instance, in a clinical setting, if you have an apparently normal-looking piece of tissue, you can tell the pathologist that this tissue is about to trip over into disease. And that has an important implication. And we have, this is what we do routinely. These are two of the microscopes I have in my lab. This is a big beast here, FTIR microscope, preparing tissue sections which we can then interrogate and look at. And again, this is another picture that we've developed, or we have. And I've got quite a large research team. I've approximately 15 in my research group currently. This has been a mixture of PhD students, MD fellows. These are people who are at senior registrar level within a clinical setting in the UK. They're doing a 2-year research project in my lab, and then they progress to becoming a consultant. I have a research technician, and as you can see here, quite a number of PhD students, and quite an international set of PhD students. So individuals from China, Brazil, etc., Eastern Europe. And I've been based at Lancaster University since 2001, and I've primarily worked in this field of environmental chemistry, environmental toxicology. And in that time, my research group has published in excess of 85 papers in peer-reviewed journals. And these have included Nature journals, they've included Environmental Science and Technology, and quite a spectrum of other journals. And I come along to a particular an industrial collaboration that I've developed over the last 5 or 6 years, and this is with Unilever in the UK. And I would say in this context, I've also been appointed to just been appointed to a government advisory committee on mutagenicity in the UK, and in certain respects, I'm advising on this particular assay. Major problem in regulatory toxicology is the application of novel assays for transformation, to monitor transformation. And there has been a huge effort to reduce and replace the use of animal experiments, especially in the application for, say, cosmetics. An assay that I've been looking at is the SHE assay, which is the Syrian hamster embryo test. And we're interested in the SHE assay because it's been around a long time, and we know that it should have a high specificity and sensitivity to carcinogens. It can truly identify with a high predictive power true carcinogens. And that has important implications because with the majority of short-term regulatory tests, if you take, for instance, the Ames bacterial mutagenicity assay, it's approximately 40% sensitive and specific to chemical carcinogens, which means you have better chance of identifying a chemical carcinogen by flicking a coin, which is actually a major thing because when an industrial company has spent several million on developing a particular chemical and it shows up as positive in a bacterial test, that's an important issue. This is a mammalian cell, 5 minutes. This is a mammalian cell. It undergoes the multi-stage process of transformation in vitro and then is stained. And what you need for this staining purpose is a trained person to look down a microscope. Now, when you stain, you can have two extremes. This is a normal culture of cells. This is a transformed culture. But you have any number of steps in between that, and that makes for a very subjective test. So what I've set out to do is develop a physical sciences platform to objectively score these particular cells and these particular cultures. And we do this by applying a physical sciences platform that can generate a fingerprint of those particular cells. Now, the problem with that is that becomes a very interdisciplinary subject very quickly because it generates huge amounts of data. If you take this spectrum here, that's not just a single line, it's an accumulation of 250 variables or features, which are numbers. If you have 15,000 spectra in your data set, you suddenly have a very nonlinear, highly dimensional and complex data set. So you need someone that can actually mine into the data and actually tease out the information. And my lab has done this over the years. We've developed in-house, with developing our own in-house algorithm scripts, the computational framework to handle that data, which then we can take a tray containing little slides where these colonies have been grown, we can put them through a processing algorithm, and we can pull out the discriminating features that are associated with particular mechanism of action of a particular chemical, or whether the chemical is non-transformed or transformed. And we can score them and give a classifier for that. And it's been hugely, it works. We can look at our vehicle control, we can compare different classes of compounds, and it generates what we think is going to be a robust technology for objective screening in this fashion. So this is something I've been working quite a lot on. I've finally been asked in my last couple of slides to horizon scan, or to predict where I'm going to be, where we're going to go in the future. I think there will be a huge need for interdisciplinary projects or consortia to allow for and crosstalk and people working for their own mutual benefit and accepting each other's language, because very often in different subject areas the language is very different. What a biomarker might be to a computational person is different to a biomarker for a biologist. What highly dimensional data to a biologist is very different to what a mathematician might consider as highly dimensional or nonlinear data. And this kind of crosstalk is difficult. But it's to allow the generation of high throughput technologies to extract biomarkers of effect. And it will require interdisciplinary science. But beyond that, I mean, how things change and evolve. I started off as a microbiologist, then I did biochemical pharmacology in my PhD, then I did genetic toxicology during my early years as postdoc, and now I'm doing a lot of work in biospectroscopy. So really, how things evolve over time is very difficult. And as one very eminent scientist once said, when he was being asked, where will he be in 3 years' time? He said, well, I can only tell you where I'll be in the next coming weeks after I know the results from next week's experiment. I think that how things change is difficult to predict. As I said, I'm based at Lancaster University. I want to also acknowledge some of my funders over the last and collaborators over the last 10 years, and I thank you very much for your attention. Thank you.